Dear students, we have to pass on to the next part of immunology, that is the interactions between antigen and antibody to produce the effect. So the antigen and antibody interactions, that is a basis for the antibody mediated immunity or humoral immunity. And the different stages that occur during antigen-antibody interactions are grouped under three headings, or we have three stages. Stage number 1, the first stage, formation of antigen-antibody complex. That is the reaction between the antigenic determinant, that is the epitope, and the paratope, the part of the antibody. Once actually they bound together, we have some visible events and reactions took place. The visible events and the reaction that take place are agglutination, clumping together, or precipitation, formation of precipitate, or opsonization, or we have neutralization. These are the visible events that occur following the binding of antigen-antibody. The next one, once all these events are actually the properties that took place, the destruction of antigen or its neutralization takes place. The destruction of antigen or its neutralization takes place. That's the final stage, the lysis of what is called the antigen, or the elimination of the antigen in the form of either microorganisms or in the form of chemicals or the toxins, etc. So these are three different stages that occur during what happens, the initiation of antigen-antibody interactions. Now it is followed by the binding between antigen and antibody. We see that one. What are the forces responsible for the binding of antigen-antibody? So the binding forces between antigen and antibody, they are responsible for all these three reactions. So the binding forces between the antigen and antibody during what is happening in the humoral or antibody mediated immunity is because of three factors. There are three factors that are responsible for binding. They are considered as the binding forces responsible for the binding between antigen and antibody. What are the binding forces? The next one, the binding forces. They also group under three headings. The binding forces between antigen and antibody. The binding forces of antigen and antibody reaction are grouped under three factors. Factor number 1, closeness between antigen and antibody. Closeness between antigen and antibody. The second one, the presence of non-covalent bonds or intermolecular forces. Number 2, the affinity of antibody. I'll explain one by one. So, what is the role of closeness between antigen and antibody? Suppose, for example, if antigen and antibody are closely fitted together, the strength of binding is great. When they are fitted together, then the strength of binding is great. If they are apart, if they are placed away, then the strength of what is called actually the binding is low. So the strength of binding is low if antigen and antibody are placed apart. And the strength of binding is high or great if they are placed together or fitted together. Just that's about what's the closeness between antigen and antibody. That's one of the factors which will decide what is called the strength of antibody antigen reactions. That's about number 1. We'll go for the second one. What are the different types of covalent bonds and or intermolecular forces? What's the meaning for that one? See, the holding of antigen, to the antigen binding site of antibody is because of bonds. These bonds are non-covalent bonds. The holding of antigen to the antigen binding site of antibody is because of bonds. these bonds are non-covalent in nature. These bonds are very weak in nature. They are normally found between the molecules, hence called intermolecular forces. What are the different types of intermolecular forces or non-covalent bonds? According to the concept, there are four non-covalent bonds or intermolecular forces responsible for antigen-antibody reactions. Now let's see it now. The holding of antigen to the antigen binding site of antibody is because of four different types of non-covalent bonds. Number one, hydrogen bonds. Now it is nothing but the hydrogen atom shared by two electronegative residues. The two electronegative residues, one from antigen and one from what is called antibody. The next one, electrostatic forces. These are the ionic bonds between two oppositely charged what we call the residues of atoms we can see. That is positive and negative. That is called electrostatic forces. The third one, hydrophobic bonds. The water forces, actually water, forces hydrophobic groups together. Hydrophobic, you know that one, disliking water. But the water molecule forces the hydrophobic groups together. That is between the antigen and antibody. The last one, van der Waals forces. It is nothing but the interaction between the outer electron clouds of two or more, actually see that one, atoms. The interaction between the outer clouds, what we call actually between the outer electron clouds, we can say a group of electrons present in the outer orbit of two or more atoms. These are the four non-covalent bonds responsible for actually holding the antigen and antibody together. This is the second factor responsible for antigen-antibody interactions, the binding force. Now the third one, factor number 3, antibody affinity. So the antibody affinity is nothing but actually the strength of reaction. The third factor is antibody affinity. A simple one. So it is nothing but the strength of the reaction between a single antigen binding site on the antibody and a single antigenic determinant. So it is a strength of reaction between a single antigenic determinant and a single antigen binding site on the antibody. So that is what is called the affinity, antibody affinity. We also have what is called ability. Ability. So affinity, the strength of reaction between a single what is called antibody binding site of antibody and we have a single antigenic determinant. The word ability refers to the combined effect of a single antibody to many antigenic determinants. Antibody strength refers to the strength of reaction. The antibody affinity refers to the strength of reaction between a single antigen binding site on antibody and a single antigenic determinant. Whereas the word ability refers to the combined strength of a single antibody to many antigenic determinants. For example, if you are taking IgM, this IgM has 10 antigenic determinants to which a single antibody reacts, and that's called ability. So affinity is different from ability. So affinity, one antigenic determinant, one what is called the paratome, what we can say the antigen binding site. Ability, one antibody, then the reaction with many what we have actually, that is antigenic determinants. Normally the non-covalent bonds are very weak in nature. As we have many covalent bonds, the cumulative effect makes the strength of binding between antigen and antibody, though they are weak. When many covalent, non-covalent bonds combine together, they provide more strength. When they are formed individually, they are less strong. When present together, they have more strength, and so that they can bind the antigen. So the antigen-antibody interactions are reversible. So to form antigen-antibody complex, then that one being reversed after the process has been completed. That's about antigen-antibody interactions, the factors responsible for, the forces responsible for antigen-antibody interactions. Now the next one, what is the role of antigen antibody interactions? The applications. Okay, now let's see. What are the applications of antigen antibody reactions? The antigen antibody reactions are being applied in different fields. Number one, to determine blood groups for transfusion, whether it is same blood type or different. To study serological ascertainment of pathogens, to infections, how far they are causing infections affecting the what is called the plasma serum. Now to develop immunoassays, nothing but the test, immunological test for quantification of substances. How much amount of substances, a particular amount of substance present in the what is called serum. That is called immunoassays, the test for the amount of substances. Then to determine the presence or absence of proteins in the serum, what type of proteins present or absent in the particular serum. For that we are using these antigen antibody interactions. We have a number of immunodeficiency disorders, for example SID. Then we have, for example, HIV is also an example for immunodeficiency disease. We also have autoimmune disease. Now, to determine the important features and characteristics of certain immunodeficiency diseases. Say an example I mentioned, we have a number of immunodeficiency diseases. This is because of the defect in the immune system, maybe genetically based or not genetically based. One such is called severe combined immunodeficiency disease, and likewise HIV. So, to determine the features, the salient characteristics of such diseases can be known with the help of antigen antibody interactions. These are the different applications of antigen antibody interactions. Okay, now let's move to what are the different just actual reactions or the functions of antigen antibody interactions. We have the following types of antigen antibody reactions. Number one, altogether we have four different types. Number one, precipitation. This is the reaction between the soluble antigen and the antibody, leading to the formation of a precipitate. So the antibody causes the precipitation of what is called normally the soluble antigen. The process is called precipitation reaction. The antibody that causes precipitation by reacting with antigen, is called precipitin. So the name of the process is called precipitation and the antibody is called precipitin. Okay, now here we have the soluble antigen. They are normally reacted with what's called the antibody. So that all these soluble antigens are precipitated together. Once they are precipitated and they are being rendered what is called harmless. So only when they are free they can work. When they are all bundled together, precipitated together, then automatically they lose their capacity to just normally create infections in the body. So this is one type of precipitation. The second type of antigen antibody reaction is agglutination. This is the reaction between a particulate antigen and antibody that results in sticking together or clumping together or agglutination of cells, and that is called agglutination reaction. The antibody which brings about agglutination or the antibody which is involved in this reaction is called agglutinin. So the phenomenon is called agglutination and the antibody involved in this process is called agglutinin. So this is a reaction between particulate antigen and agglutinin. Particulate antigen means either the cells, the RBCs, the bacteria, viruses, anything else. Here you see that one, the bacteria, which are clumped together, stick together, so that what is happening, they cannot do the work. They cannot create any infection. So they are being normally just rendered harmless by the immune system. Now the third type of this antigen antibody reaction is opsonization, otherwise called enhanced attachment or coating over. Now what is happening? When antigen antibody reacts, that results in marking of ingestion and destruction of pathogen by phagocyte. The meaning for that one, now the pathogen or the bacteria are just normally processed so that they can be easily phagocytosed by what is called the phagocyte. That is called the marking. Antigen antibody reaction that results in marking of just it gives actually the phagocyte to identify the pathogen, bacteria, and the marking. That results in ingestion and destruction of pathogen by the phagocyte. Then, the entire body of bacterium is coated over by a substance what is called opsonin. The antibody that is involved in opsonization is called opsonin. It binds to the receptor of the pathogen cell membrane. Now this is the cell membrane. This is what we have actually, the cell surface receptor of the pathogen. It reacts with the antibody, covering. So many antibodies are attacking, and each antibody binds to what is called the cell surface receptor present on the surface of pathogen. So after binding, what does it normally do? The opsonin attracts the macrophages and neutrophils, they are nothing but the phagocytes, towards what is called the pathogen. So that the macrophages and what we call the neutrophils acting as phagocytes do the process of phagocytosis. That means this opsonization facilitates the phagocytosis of pathogens by the macrophages as well as the neutrophils. Okay, so what is the meaning for that one? So opsonization is a process in which the pathogen is coated over with the substance. You see that one entire pathogen is coated over with a substance, and this is what is called opsonin. This is opsonin. And this is what is called cell surface receptor, to which only the opsonin is binding. Many antibodies are binding to many what is called the receptors of the pathogen. So it's a process in which the pathogen is coated over with a substance called opsonin. Entire body is covered. And marking pathogen, just actually shows or reveals the pathogen out for the destruction by phagocytes. The marking is nothing but exposing the antigen to the processed antigen to the phagocytes so that they can do the process phagocytosis easily. And that is what's called the marking. This results in much more efficient phagocytosis because of this what is called. Easily the phagocytes engulf the processed bacterium, the opsonized bacterium, because of the coating over by the opsonin antibody. So this is normally resulting in what is called phagocytosis with the help of what is called antibody molecules. This is the third method or third type of antigen antibody interactions. The next antigen antibody reaction type is neutralization. Once again, it is an antigen antibody reaction that results in elimination of harmful effects. Harmful effects of what? Bacterial exotoxins and then viruses. Neutralizing the exotoxins released outside by the bacterium and also the viruses. Destroy the viruses. Now the neutralizing substance, that is an antibody, is called just what is called antitoxin. The antibody here which is involved is called antitoxin. Okay, so we have agglutinin, precipitate, opsonins, antitoxin. So the neutralizing substance, nothing than antibody, is called antitoxin. So this is produced in response to what's called the bacterial exotoxin or toxoid, the inactivated toxin, by what is called the antibodies. By what is called by the antibodies, or we can say by what is called specific immune cells. We can write in this way also, by specific immune cells. We can write either antibodies or specific immune cells. Either way you can call this one, I am writing the same one, antibodies. or specific immune cells. That's about neutralization. So the antibodies are called antitoxins. The process is called neutralization, that is involved in neutralizing what we have, the bacterial exotoxins or the viruses. That's about the fourth type, neutralization. It's also a kind of antigen antibody, that is interaction or reaction. Now all these four types are involved. The main aim is phagocytosis, either precipitation or neutralization or agglutination or what we have opsonization. All these events are mainly involved ultimate effect phagocytosis. Now we have one more, just what is called a complement system. What is the role of the complement system? What is a complement system? What is its function? Just a few points about this one. What is complement system? Now the complement proteins and their activities together called complement system. What are complement proteins? Now these are nothing but inactive plasma proteins or cell membrane proteins. Inactive plasma proteins are cell membrane proteins. During infection, these proteins are activated to form a complex. What complex? Membrane attachment complex. It is attached to the surface of the infected cell and causes normally holes or pores in the membrane. So that what is happening, the fluid from outside, the extracellular fluid, ECM, the extracellular fluid, enters in the cell. So that what is happening, the bulging of the cell, ultimately the bursting of the cells occur. This is called cell lysis. So it causes the cell lysis of the infected cells, a mechanism brought about by complement system. It is also involved in inflammatory reactions, that is called inflammation and phagocytosis by means of coating wall. What is inflammatory response? At the site of injury or infection, there is a pain, a redness, swelling and generation of heat or fever. All these activities together called inflammatory response. So at the site of injury or infection, there may be swelling, redness, pain and generation of heat. And all these reactions together called inflammatory response. It is also one of the defense mechanisms coming under innate immunity, one kind of barrier, what's called inflammatory barrier, that is brought about by what is called the complement system. That's about the complement system. So the complement system is involved in inflammation, cell lysis and also phagocytosis. The previous four, they are involved only in phagocytosis, but this one is involved in three important processes. 1. Inflammation, 2. Phagocytosis and 3. Cell lysis. I showed that one in the picture, a simple picture, within 2 minutes. As I described earlier, the complement proteins attached to the cell surface, this is a cell membrane, the molecular structure. It creates a pore. Through this pores, the ECM, the extracellular fluid, enters, causes the bulging and bursting of the cells leading to cell lysis. This is one. Now it also involves just inflammation. At the site of injury, there may be redness, swelling and pain. Generation of heat, called fever. All these reactions together called as inflammation. So it is also just created by the complement system. So the complement system has 3 functions. 1. Phagocytosis, 2. Cell lysis and 3. Inflammation. These are all the 3 functions of the complement system. This is the mechanism of cell lysis. Creation of pore, entry of fluid, bulging of the cell, bursting of the cells, leading to the destruction of the cell, that is called cell lysis. In the next class, we have to discuss something about vaccine and immunization. Okay, that's the next part of this lesson. what are the different types of vaccines, and also the immunization and vaccination. So I will just meet you once again in the next class. Till then, goodbye. Thank you.